Let me give you, I don't know if you guys, I don't think we talked about it in any of my periods except fourth period. What about a toxin that caused excess potassium to be outside the cell? So the outside of the cell gets flooded with potassium. Remember, potassium is normally pumped in thanks to the sodium-potassium pump and then leaks back out. What would be the consequence of flooding the outside of the cell with potassium?
Would it be harder to shoot, like, what's it called, shoot up?
The rising phase?
Yeah.
Let me think about it. No, it would actually make the rising phase more likely.
The resting would not be -70.
What would it be? Like more positive or more negative?
Negative? Positive?
Okay. Think about it. Potassium is normally diffusing out from the leaky K. If we make the outside environment really, really concentrated with potassium, that reduces the high to low concentration gradient, right? Normally it's high potassium on the inside, low potassium on the outside, and that ensures that potassium leaks out, right? But if you take the outside and make it super concentrated with potassium, is the potassium gonna leak out?
Not
Is
as
it gonna
much.
diffuse down? No, right, not as much, because it's not, the concentration gradient isn't there. We're destroying the concentration gradient. So if positive charges aren't leaving, that means the inside of the cell becomes more positive, which means it's closer to threshold naturally. And that could be dangerous. That could cause, for example, seizures. What about one that causes the membrane to become naturally hyperpolarized? What might the toxin be doing there?
Hyperpolarized?
Hyperpolarized just means naturally lower than resting, which is -70. What would be the mechanism of action? Okay, something that causes, maybe something that makes the leaky potassium channels open more, or more of them to open, so more potassium leaks out. Yeah, sure. Or maybe yeah, exactly. So what would be the consequence here? Neuron more difficult to fire or less difficult to fire?
More difficult.
More difficult to fire, because it's more difficult to reach threshold. What about a toxin that does something like this? What's happening here?
The calcium or
Not
the potassium won't flood back in or flood out.
flood out, right? Potassium's not flooding out, which means what's probably not working? The potassium voltage gated channels are probably either not opening, or if they are opening, they're blocked. So the inside is going to get flooded with sodium, and it's not going to get negative again because potassium isn't rushing out. I mean, I can't think of any more.
Oh.
What about something like this?
The leaky K doesn't work.
No, we know that the leaky K works because the resting is still -70. So the leaky K is working fine.
I thought it was the resting.
The voltage gated channel, maybe it's not letting as much out as it usually does.
The voltage gated potassium channel may be closing prematurely. Yeah, it closes faster than it normally does, maybe because of how a toxin interacts with the protein. And also remember that voltage gated sodium channels normally don't respond to -50 during the downfall. They only respond to -50 during the movement of resting to -50. So there's something wrong going on here where the voltage gated channels are sort of like hyperactive, they're overactive. And because of that, they're opening more than they should be. So that could be an answer. Both of those explanations would get you credit. There are multiple things that can be going on here. Let me see if Mr. Barnett needs help or needs to do something. Hey, what's up? Yeah, I put it in the back of your readings. The solutions are already made.